Okay. This is, we're going to talk about speciation. How does it happen? How do new species come into being? The most important thing to understand is there has to be a lack of gene flow between two different populations. Now, that can be caused by a geographic barrier. It can be caused by behavioral. We're going to go through that. But for two populations to diverge that were at one time one population, there has to be no gene flow. And then the forces of evolution work on each population separately, including genetic drift, mutation, non-random mating, and then, of course, evolution by natural selection. Okay? Now, this is just a bunch of different examples. Those are the Hawaiian, let's see, up here. These are the Hawaiian honey creepers. One, probably a couple of individuals of one species came from Asia 5 million years ago, landed on probably Kauai or something like that. And from there, as different islands arose in the future, individuals dispersed and new species were created because there's very little gene flow between the islands. And the islands also have very diverse habitats. Okay, so species they come from a common ancestor. And then, of course, there's mechanisms of speciation, and that's what we're going to get into. Okay, we're going to talk about what is a species. I know one when I see one. These are all considered species. But it turns out there's a whole bunch of different definitions of species, because species aren't actually real in the sense that, like H2O, like water is real. Water is two hydrogens and an oxygen. A speciation is an ongoing process, not like, oh, at this point you're a species. It's not quite like that. But it's not arbitrary either. It's not like you take animal of various sizes, let's say 0 up to 20 feet tall, and you small, medium, and large. That's arbitrary. Species aren't like that. They're semi-real, but they're not as real as like H2O. But we have different definitions of species, and we're going to learn 3 of them. And they're good for different things, as we'll see. So, species, for the law at least, is the smallest evolutionary independent unit. That implies there's no gene flow, but that mutation is happening, selection is happening, gene flow and drift operate on populations separately. Okay? So, if you split 2 species, one's over here, one's over there, there's no migration back and forth. Each species is going to be now on its own separate evolutionary trajectory. So, and then this is the main definition. It's called the biological species concept. Species consist of interbreeding populations that evolve independently of other populations. So, lack of gene flow, species form a boundary for the spread of alleles. So if there's no spread of alleles, then these populations are on their own. And, well, I already did that bit. So these are considered species over here. So, come on. A lion and a leopard. Okay? The lion is quite a bit bigger. But this is our favorite animal, the dog. And you can see how different they look in terms of size. well, it turns out there's one gene that actually is expressed differently, and that's why you have different sized dogs. But so there's great variation in some species, and in others, there's not much. Now, of course, for dogs, we've been breeding them, artificial selection, for thousands of years. Okay, so we have all these different breeds. Okay, so there's three species concepts we're going to use. The first is the morphospecies concept, that morphology. Or we can call it a phenotypic species concept. Species are groups of individuals that are morphologically similar. They look the same, clearly distinguishable from individuals in other groups. The trouble is that it's kind of an opinion. Oh, this is a species and that isn't. There isn't definitive criteria. You can have big geographic variation in morphospecies, and so they look different in different areas, but they're the same species. So that's a problem. There's things called cryptic species. They all look the same, but they're actually different. The thing is, morphospecies are the only species concept we can use for fossils. Okay? We can't use, well, occasionally we've been recovering some ancient DNA, but we'll get to that. But for the most part, fossils, you can't get DNA out of them. The biological species concept. A species is a group of actually or potentially interbreeding organisms that can mate and produce fertile offspring. Okay? So viable offspring and fertile, that means they're not sterile, they're not like a mule or something. Biological species concept, we don't know with fossils whether they are actually breeding or not. I mean, we don't have direct evidence of that. This concept doesn't deal well with hybrids, you know, where there's two different species that hybridize and might even actually create viable hybrids. Okay, so there's some problems with it, but it is the one that we use most of the time. And then if we look back in time, and historically, we use what is called the phylogenetic species concept. Anybody remember what a phylogeny is? Remember we just saw one with the 3 domains? That's a phylogeny. It shows evolutionary history. Okay, here, okay, so this is a phylogeny. We have these different salamander species. They all share a most recent common ancestor down here at this node. Okay? The first species from this group, from the ancestor, was A, and then there was this other clade over here, B. It wasn't like A and B, it was actually A, and then A is sister to this whole clade of A and C, B and C, okay? So these show branching patterns and relationships, evolutionary relationships. So that's what a phylogeny is. And we usually phylogenies show time. So this is old and this is newer, okay? So, phylogenetic species concept? Yeah.
Is there a difference between a histography and a phylogeny?
What?
Histography? I thought that's
Histography?
yeah,
No. Phylogeography, we'll get to that. Historiography or something? No. Oh. I'm not sure. That doesn't sound so
I
right.
something else.
Yeah. So, there's different kinds of phylogenies. There's cladograms, phylograms. Okay, but for now we're just dealing with straight old cladograms. So, it's the smallest set of organisms that share an ancestor. Okay, so here we are again. Okay? And so here's the ancestor to all of them. Here's an ancestor right here at this node for species B, and then this group of species that actually are ring species in California and Oregon. If you go up the coastal range, you'll find these different grades of salamanders that look kind of the same but different. And then you go around up into Oregon, and then you go down the Sierras. Now, each of these different populations can actually interbreed, so they're separate species. They're not separate species, they're considered one. But, so this can breed with this can breed with that, and so on. The trouble is, this probably cannot breed with that, and certainly this down here cannot breed with the original population. Okay? So originally this species came into, this whole group of species came into being because of a geographic event. Okay? And then and they came in from the north, and then they kind of went down, so they shared our most recent common ancestor. But because they're separated, not complete gene flow, but mostly, you end up with the actual, some things are species and some aren't. But the, in terms of the phylogenetic species concept, we have a common ancestor, and then we have these descendant species, B and this C complex. Okay. Well, don't worry about the problems with this. It's more of a historical level. So, the biological species concept states that species are defined by who they can actually successfully breed with. Are you my species? Well, that little guy is probably, I would rather not find out, right? But all dogs are the same species despite considerable variation. Now remember, morphospecies concept, you primarily use that for fossils. Phylogenetic, you use that looking at evolutionary history. It's a look back in time. And you can make a phylogeny out of DNA, out of fossil characters, anything like that. Mostly we use DNA now. Yeah?
I have a question. So the species concepts, those are all just theories on like what the definition of the species is?
Yeah. Yeah. That's why I say that species aren't real.
Yeah.
So if you, like water, we can actually define that as 2 hydrogens oxygen, right? I mean, that's what it is. Well, speciation is an ongoing process. It's not like, oh, now it's a species. It's not quite like that. Okay? So usually when people are looking at speciation, they're going to define which concept they're going to actually use. Okay, so everybody's on the same page. So speciation is a 3-step process. Gene flow is disrupted between populations. Populations accumulate differences in alleles, and eventually reproductive isolation evolves. That means they can no longer breed together either, and we'll look at that. So kind of a schematic on how it is. We've got, this is one population right here, okay? Might be, and this is time. There's interbreeding between all these maybe subpopulations. But at a certain point, there's a barrier to this population. So let's say a mountain range rises up, and obviously it happens over many, many years, millions of years, but suddenly the population is now split into 2 different populations, and there's no more gene flow. So that's a geographic barrier. But there can be other kinds of barriers. As long as reproduction is interrupted and there's no more gene flow, then speciation can actually happen. Okay? Now, Um, there's also different modes of speciation in terms of geography. And let's take a look at this. Okay. So this is allopatric. Allopatric means other land. So in other words, two different populations in different places that were at one time one population. And the barrier is usually a large barrier. So here's a population, and then there's some barrier. It's usually a geographic barrier, and it's called vicariance. So vicariance can be a mountain range rising up, a stream separating two different populations, being on different continental plates. And so over 300 million years, if you actually look at what the continents looked like 300 million years ago, it was Pangaea, one big one. There were two parts to it. Then Gondwana and Laurasia separated. Gondwana then separated eventually over time into Africa, South America, Australia, New Zealand, India, and let's see, what else? Oh, Antarctica. And there were actually things living on Antarctica back then. Okay? So that's vicariance. Now, you can also have allopatric speciation. Do you guys remember what a founder event is? Okay. So you have a main population, let's say a mainland island model, and a small group of individuals dispersed to, let's say, the new habitat, the new island. And then there's very little gene flow between the two after that. Well, first of all, the population that disperses probably doesn't have a full complement of all the alleles found in the actual population. But there's still no more breeding going on, localized colony. They start diverging because of different selective forces, genetic drift, small population, drift is strong, large population, genetic drift is weak. Okay, and then, let's say you have range expansion and then they come back together. And at this point, one of two things can happen. They can actually start breeding again and make viable hybrids, and then they'll coalesce back into one species. Or by this time, there's such a great difference between this population and this population that they can no longer breed anymore. And the same thing goes here. So let's say you have this mountain range, and eventually over time, it's ground down, erosion, and then these two separate species now come back together. And again, they might form like a hybrid zone, or they might stay completely a separate species. And then there's one other, so instead of allopatric, there's something called sympatric speciation. And sympatric speciation is in the same place. Okay, and if we actually look at this, these are, there was all one population right here. They all look the same. But somehow in the habitat or the area that they're actually breeding, there are habitat differences, and they start segregating in terms of habitat. So we're going to look at an example where there's these flies that are that infest hawthorn trees and their fruit. Well, a relative of hawthorns in North America is apples, and they were brought from Europe. Eventually, some of the flies started actually landing on the apple trees, and they got attached to the apples. The scent, that's where they wanted to breed. That's where they were actually born. And so eventually you start having the population, even though they're in contact and there could be gene flow, right, between all these separate things, eventually you end up with two populations that aren't breeding anymore. Okay, and we'll look at that right now. Okay, so this is an example of vicariant allopatric speciation. This is the different color morphs. So we've got light green, dark green, brown, black, moderate green, whatever. And they're all 5 different color morphs. They're all at 20% of the population. Okay? Now, there's this big river here, and things are going to happen. The river changes course. It now splits these populations in two. They still start out with what they do is they start, because they've been split, they start to diverge. There might be soil types, let's say, that exert selection in terms of being cryptic. So if you're a salamander, you don't want to be seen, so you have color morphs according to the soil type, let's say. And then over in this group, eventually, there's a mutation. And these spotties actually are even better at hiding in the background than the others. And so they're going to start proliferating, and the actual morph percentage of each morph starts to change with the population. This is 40 generations later. There's more spotties, okay? And these proportions are changing as well. And this is an example of allopatry. The Isthmus of Panama, guys are probably familiar with it. Okay. Now, if you go back 5 million years, the Isthmus of Panama wasn't there. Okay? North America and South America were completely separate. And so, on either side of the isthmus, well, this was all ocean, right? And these different types of shrimp were actually all, they were a couple of different species there. So maybe this guy and that guy up there, they were actually identical to each other, but in the last 5 million years they started to change. And so there's these sister species across taxa. Now when there's sister species now, because the Panama Isthmus rose up and then separated the whole population, because now these things live in the ocean, okay? So you can actually look at snapping shrimp. There's, let's say, Pacific 1 and Caribbean 1. Let's say up there and this one. At one time they were one species. They're still more similar to each other than they are to any other snapping shrimp on either side, okay, because they shared an evolutionary history. And the same thing with all these different species. So this is an example of allopatric speciation. You've got a barrier that interrupts or in a continuous population, and now those populations are separated. Now, you could also get the same sort of thing, but now we're having dispersal. So we're going from this large population, there's a little bridge, let's say like the Bering Strait during the Pleistocene glaciation, water level dropped, species came across, then eventually the water level started rising again, and now we've got a population that is in fact separated. And it's another, let's say, probably a founder effect. Okay? And again, you have a spotting mutation, And it's probably selection for each different area in terms of the soil or something like that. And so eventually these might be two different species. And this is after, let's say, 60 generations or something. This is a good example of dispersal and colonization. The Hawaiian Islands, they're really out in the middle of nowhere in the eastern Pacific. And it's amazing, these are oceanic islands. So they've never been attached to a mainland at all. And somehow, these different species made it, small founding population made it to Hawaii. Okay? Now, there was Drosophila, these are fruit flies. At one point, some Drosophila probably got blown thousands of miles and landed on Kauai, because that's the oldest island there. And then, eventually, Oahu, volcanic activity, later the whole Maui bunch, Molokai and all that, and then right now we're with Hawaii, and then over here there's another island that's starting to form. Well, these fruit flies dispersed from one island to the next. And there's a fair amount of distance, so there was a lack of gene flow, and they started to differentiate. Well, also, the Hawaiian Islands, if you've ever been there, there's a lot of topography in terms of, there's lush valleys, there's high volcanic mountains, all kinds of different habitat. So there was strong selection for that. So now there's about, I think, over 500 species of Drosophila, but they're all related to each other because they had a single most recent common ancestor that somehow made it to the Hawaiian Islands. Silver swords are a kind of plant. They probably rafted over from North America. Their closest relative is in North America. And then there was crickets, and then these birds. So, this is an example where dispersal, founder effect. And that, in fact, each of these events resulted in a proliferation of new species on these islands. So if they didn't stay just one species, they proliferated into many different ones. Okay, so here's sympatric speciation. It means in the same place. It means there's still some gene flow here. And so if there's gene flow, there's not isolation. So how can it actually happen? For a long time, people said it didn't happen. But there are some examples. And we often see this with insects and the plants that they eat. Plants protect themselves with different toxins. The insects, there's this ongoing arms race going on between the two of them. And eventually you've got populations that are best at, let's say, eating one kind of plant because they can deal with that toxin and not another. And so you end up with a disruptive selection where even though they're in the same place, these populations are no longer interbreeding with each other. So this is the hawthorn and the apple tree. And the flies, there was initially only hawthorn trees and these hawthorn flies. And then they brought over apples. And they were in the same area. They planted them next to hawthorn trees. Okay, all in the same place. Well, over time, apple flies, they started, they were born on the leaves of apple trees or on the fruit. That smell, they like that smell. So anytime they're presented with that smell. So you can say, how did it happen? Well, you could propose that, let's say, they fixated on the smell. So you could take these flies into laboratories, which is what they did, and they actually did an experiment in putting a fly in and then two different scents. And depending, you would predict that an apple fly would go to the apple scent and the hawthorn the same way. And that's exactly what happened. Okay? So this is one of the few cases where sympatric speciation might happen. Okay, now we've talked about reproductive barriers. There's different kinds of barriers. We've talked mainly about geographic barriers, but and so we might have spatial isolation, like we've been talking about. Here we've got two different spotted owls. The northern spotted owl, which is up here, the Mexican spotted owl, which is here and here. Now, at one time, they were all one species. Then the climate changed, things dried out, and they started specializing, in one in a desert habitat, the other in the Pacific Northwest. Okay? So that's, and the ecology, the environment they were in was different at this point. You can also have, so that's spatial isolating mechanisms. This is what we've been talking about, vicariance. But you can also have temporal isolation. These are corals. The same species of corals release their sperm and eggs. It's a highly choreographed thing where they all do it. Each species does it at a different time, but all together. Okay? So that, you know, you're not wasting sperm and eggs that won't get fertilized. So in this case, there's temporal isolation between the different species. And they don't breed anymore. Okay. There's also something else called behavioral isolation. So these are blue-footed boobies. Probably like that name. They look funny. They have this very intricate courting thing. So what happens is, after they've bred, they all go off on their separate ways. And then they come back, I don't know, about 8 months later. And to reestablish a pair bond, they go through this whole courtship dance. And they continue to breed like this over and over, where they're apart and then come back together. Well, if you had different species, they would have different courting dances. So the female of one species would not recognize what the male of the other one is doing. Okay, so this is behavioral reproductive isolation. They just don't recognize that they're the same, they're different species, okay, just because of the behavior. Now this is all called prezygotic, and what that means is before you actually have fertilization, okay? So you can have also post-mating but still prezygotic, like these different monkey flowers, and the way they're actually the insects that actually pollinate them are different. They bloom at different times of the year. Okay, so there's also an idea where it's mechanical, so they don't. The different insects actually don't get pollen from one and bring it to the other. Okay. You can also have it where, just think about those two dogs, the big one and the small one. Well, think about it instead of an elephant and a mouse. How are they going to breed? There's mechanical problems there, right? Just a little bit. And then there's also what's called gametes, like sperm competition. Okay, but don't worry about that. Then, and then there's post-zygotic. That means after fertilization. And a lot of times you end up with an embryo that's inviable. So it's a hybrid between two different, like goats and sheep and goats. They have different chromosomes and numbers. They'll breed, they'll form a zygote, but it's kaput after a certain developmental period. It doesn't produce any offspring. You can also end up with sterility. That means they're going to produce a viable offspring, but they're not viable in terms of actually reproducing. So it's a dead end. And in this case, Horses have 64 chromosomes, donkeys 62. In terms of egg and sperm, the horse egg has 32 chromosomes, the donkey sperm has 31. There's a problem, they end up with a mule that's sterile in general. Occasionally, you'll get, depending on which one is the male and female of whether it's a horse or a donkey, you can get a jenny instead of a male mule, let's say. You get a jenny that's actually fertile to one degree or another. But it's pretty much of a dead end. So this is after they've formed a zygote. One cell, it starts dividing, and then changing, so you have different body parts and all that. And if things don't work out right, you either have an inviable embryo or you have one where you have an actual organism, but it's incapable of breeding. Okay. Biogeography. Why are certain organisms found where they are? It's a big question. This is Wallace. He is the father of biogeography. And it turns out, so how did they get there? Well, a lot of times, all these different species in this one area are related because they descended from a single most common ancestor a long time ago. And so why is it that in Australia, you have all these marsupials, and then almost everywhere else, placental mammals? Well, plate tectonics. Continents moved around. Australia split off before placentals even arose. Okay? And so in Australia, you have all these different marsupial mammals, kangaroos, possums, bandicoots, all sorts of different things. Well, there's this line right here. It's called the Wallace line. So all the species in this area are similar to all the species in Southeast Asia. Okay? And then on the other side, it's Australasia, including all these different marsupial mammals. Okay? And then the other side, they're all placental mammals. This is because Australia and then Laurasia have been split apart a long, long time. So each has its own fauna and flora. Fauna is animals, flora is plants. Okay? So, and you have different kinds of distributions. Endemic means the small population in one area, cosmopolitan all over, continuous versus disjunct, but I don't think you have to worry about that. So we have these biogeographic regions that are different, and how did they get that way? So part of it's historical. Past processes that influence current distributions, plate tectonics, or dispersal. Something dispersed to a new area, that's how they got there. And then you have origin of new taxa, the vicariance. You also have extinction going on. Some populations wink out of existence. And then in terms of contemporary processes that are ongoing, that were ongoing in the past as well, well, there's different ecologies. Different kinds of environments have changed over time. We're no longer in a glacial period. We're in a long warming period from about 10,000 years ago on. So different distribution in terms of climate. And then there are species, you don't have one species that can live everywhere. Doesn't exist, right? We're pretty good, but actually some insects do better than us. But you have two different ecological differences, abiotic. So physics and chemistry. salinity and differences in salinity in water. Temperature, changes in different temperature, so some species can tolerate warmer temperatures, colder temperatures, that sort of thing. Percent of oxygen if you go from lowlands up to a high mountain peak, those are physical, that's abiotic. And then there's biotic interactions, like interactions between an insect and a plant that it pollinates. That's a mutualism, or this predation, one animal eating another. Herbivory, one animal eating a plant. So these are biotic interactions. Actually look at this on your own on the Moodle site, but it starts out with where the different plates and continents were about 400 or 500 million years ago and how they changed. So this is called paleogeography, how geography has actually changed over time. So you can end up with organisms that might be related to each other or look like each other in two ways. One is they shared a most recent common ancestor. They inherited the traits, just like we inherit our traits from our parents, right? Or you can end up with something called convergent evolution. Convergent. Now, convergence has nothing to do with the same ancestor. It has to do with widely divergent taxa, but they evolve similar characteristics independently of one another due to similar selective environments. All the animals that fly, bats, birds, insects, they didn't all have one common ancestor and then they all inherited flight. You can actually look at the structures of insects and bats and all that. They each independently evolve flight. Okay? Now, it's because flight, you've got an environment where you can actually travel wonderful long distances and you can fly around. That's great. It enhances individuals. Oh, yeah, we'll play with that later. And so, flight is a convergent evolution. In other words, it evolved independently in different taxa. A lot of insects, taxa had flight, and then they lost it. Snakes, they had an ancestor that had 4 legs. They lost their legs because of the kind of environment they were in. Having no legs was better, or higher fitness. So convergence, you can have the same traits that look very similar, but has nothing to do with inheritance. Whereas phylogeography, phylogeny, that's ancestor-descendant relationships, and we do that with a phylogeny. And you can actually take a phylogeny and overlay it into geographic areas and show how things got where they were. Either the continents moved around, so you had this group over here, this group over here, you had dispersal. You can do this kind of phylogeography that's going on. So here's a good example. About 350 million years ago, we had Pangaea. It was one large continent. There were 2 different parts, Laurasia and then Gondwana. And you can see this is what it looked like. And they shared taxa. So that's why you can find fossils, very similar fossils in the same strata in South America and Africa, because at one time they were connected, right? And they split apart, but the same fossils are still there. And the same rock types are still there, okay? So here we have this Cygnathes, a Triassic reptile. was found throughout this area. You can find the fossils for this in South America and Africa. Let's see, this is a plant that was found all throughout Pangaea, including India, Antarctica, Australia. There are probably descendants living there right now. Yeah. Is it time?
Oh, she has a question.
Oh, yeah.
On the last slide, so is convergence, like, I don't know how to say it, homoplasy? Like, remember the dolphin slide?
Yeah,
It's the same
that's
thing.
exactly what it
It's
is.
just for, like,
So if you were to use, let's say, flight as a character, and you created a phylogeny based on that one character, you would, let's say, put insects and bats and birds and all at one, with one common ancestor. Well, that's not how it is. That would be misleading. That's homoplasy, right? Good question. So we'll look at the dolphin thing again. Dolphins and plesiosaurs. They look like dolphins, but they were actually reptiles during the Jurassic period. They both look like the same because they have to move through water, and there's a fusiform body that does much better in some circumstances. You know, instead of being a block of wood moving through water, doesn't work so well. Okay. So this is vicariance. There's also these flightless birds. They all started with one common ancestor. It looks like probably tinamous in South America. The continents spread apart, and now you've got rheas in South America, ostriches in Africa, a couple different species, emus and cassowaries in Australia, and kiwis in New Zealand, and also an extinct group called moas that were huge birds, like 10 feet tall. But they got wiped out by the Maori tribe once they colonized the area. Extinction. So we have these 6 major biogeographic regions. We have the Near Arctic and the Paleoarctic. Now, at one time, this was all one landmass. So Near Arctic and Paleoarctic share many species, like gray wolves, brown bears, grizzly bears, all sorts of different things. Okay. Then there's the Neotropical in South America. South America has a bunch of very strange animals. They also have a huge diversity of birds. The West Indies, each little island is separated, and you have evolution occurring on each one separately. Then you've got Africa, the Ethiopian, Madagascar, and then Mediterranean up here, up in North Africa, and the Mediterranean region. Oriental, here's the Wallace Line, Australia, New Guinea, all that. Okay, so you have different kinds of taxa in different places. Whether it's plants or animals or fungi, whatever it is. Now, of course, you can also find taxa that are related to each other, but they're in separate places because they disperse. So, for example, seed dispersal. Seeds can be carried for hundreds or thousands of miles by different kinds of animals. Or they can be carried by wind, by water, all sorts of different things. So obviously plants can't pick up and decide to walk over somewhere else, so they have their babies do the dispersal. They say, go forth, prosper. Most of them die. And pollination is the same sort of thing. Then you also have kind of vicariant events, as we've talked about. There was this great American interchange. Once the Isthmus of Panama rose up and connected South America and North America. Each continent had completely different taxa. some of the taxa actually moved from South America and colonized North America. It turned out it was asymmetrical. A lot more the colonists went from North America to South America. They were just better. A lot of these, some of these were marsupials. This was an old glyptodon that looks like an armadillo, but it was huge. It was like 6 feet tall or something. Giant ground sloths, all sorts of different things. So starting about 5 million years ago, you had all these different species going across the land bridge and colonizing different areas. Another good example is hummingbirds. They started out in the Andes in South America. Over time, they proliferated into like 330 different species. Almost 90% of them are still in South America or Central America. And then the farther north you go, the fewer species of hummingbirds you actually have. Okay? So you can see species distributions depending on the evolutionary history, either because this is where their ancestors grew and then had descendants, or where you have groups that then disperse to new locations. So this is an example of convergent evolution again. These all look like cactus. This is cactus, as a matter of fact. But this succulent here is not, the cactus is only found in North America. Why do they look the same? Where does cactus live? What kind of habitat? Desert. Okay. Same thing in India. There's huge areas that are really hot, dry deserts. Okay. And then Africa, the same sort of thing. Euphorbia, not at all related to each other, but looks the same. So this is because you have a desert environment. The solutions for surviving in a desert environment, there's only so many solutions you actually have. And so you get using different taxa, using the same solutions, but they're not related to each other. Nectar feeders, in terms of these birds, and then this is actually a moth. This is Hawaii, one of the honey creepers, and they pollinate flowers, and they also, flowers have nectar, they come drink it, go off to another flower, pollinate it. Hummingbirds, they do the same thing. This one's from South America. And then there's this moth, called a hummingbird moth. They do the same sort of thing. Both species actually hover when they're feeding. Okay? And then this is a sunbird in Africa. But you see similarities because they're all feeding on the same food type. And you need specialized structures to actually feed on nectar. So here's our convergent evolution again, homology. Up on top, these are the Hox genes. They're developmental genes. All animals except, we're almost done. All animals except the sponges have Hox genes. In this case, these are fruit flies. And then down here, we have humans, they're vertebrates, right? Well, they have the same Hox genes, right? Same color, same genes from the same ancestor. Okay? So even though fruit flies look like very different compared to humans, we still use the same genes in terms of development and body plan and things like that. So you can actually see the phylogeny here. We've got this chordate phylogeny. Here's the most recent common ancestor. You have these two different groups of animals. One group includes the insects, the worms, and mollusks. and the other sea stars and sand dollars, and then chordates, including all the vertebrates. Now, this is convergent evolution on the bottom here again. We saw this earlier. Common dolphin, and then ichthyosaur. Look very similar. Well, they both live in marine environments. They probably both ate the same kind of food. They were chasing fish and stuff like that. They have similar body types because they filled the same niche. In other words, they lived the same sort of life, did the same sort of things as each other, and so they ended up looking similar. But this is a reptile, a seagoing reptile, and this is a seagoing mammal. And they each lost their limbs at separate times. Okay. Now, an adaptive radiation, you have, let's say, one species, and it proliferates into a whole bunch of different species. Lots and lots, like the Drosophila, the fruit flies in Hawaii. There's over 500. They started with one species. Okay. Now, normally, it's hard for speciation to happen because there's incumbent taxa. That means there's taxa already living there, probably doing the same thing you're going to be doing, but they're better adapted to actually doing it. Okay? So if you're going to actually displace an organism from one place, it's a very hard thing to do. But it's called competitive displacement, and it can happen. But there's another thing that I want to talk about. Is called a niche. You've probably heard about a niche. You know, a niche is like a lawyer, or a doctor, a mason, a farmer, kind of different ways to make a living. And a niche also has to do with perhaps different kinds of habitat. So you have specialists, let's say, that live at high altitude compared to lower altitude. They have different tolerances in terms of temperature. So that's what a niche is. Now, an adaptive radiation, you actually need opportunity. And there's kind of three ways that you can actually get it. You can get to a new environment where there aren't very many other taxa, like Hawaii, dispersal. You can have a mass extinction where everything gets wiped out but a few things, and suddenly everything's wide open. And then you have a proliferation of species after the extinction event. And you can also have what's called phenotypic innovation, like feathers. Feathers arose initially for insulation. So we actually have dinosaurs with like downy feathers. We also have dinosaurs with kind of body feathers with color, so they were used for display. Same sort of thing. And then eventually, they were used for flight. Okay? So when you have evolution of something entirely new, it opened up a whole new realm for species to proliferate and live in. So extinction and speciation is just like what happens with birth and death. Right? Oops, he's dead. That's extinction. Oops, they're dead. Okay. Same sort of thing here. These are type of dinosaur. These are some other ones. They all went extinct. Here we have taxa, descendant taxa that are still with us. Okay? They start at different times, so the birth of different species. Lineages, they start out as separate species, or as one population, they get split, and then over time, you get all these groups like felid, cat, members of the cat family proliferating. Okay? So that's like the birth of species. And then the death of the species is extinction. There were 5 mass extinctions. The biggest one was the end Permian extinction, 250 million years ago. About 90% of all species on the planet were gone. It took about 30 million years for things to start coming back. Remember, there's whole groups of taxa that are no longer there. Trilobites are gone. These marine things that they were really very, very common, all kinds of different species. The Permian extinction, that was the end of it. They were gone. The same thing with the Cretaceous, the KT, that's the one that killed the dinosaurs. So there's no dinosaurs left except for birds, right? That's the only ones that are left. It took about 10 million years for that to, reconstitute. That's when you actually had adaptive radiation of mammals, because there are all these niches that previously were occupied by different species of dinosaurs. So here's the asteroid. They actually found the crater off the Yucatan Peninsula. There's shock quartz that you get from a big impact and microtektites. There's this iridium, which is a chemical that's only found mainly on asteroids and very rare on Earth. There's this layer at 65 million years ago, worldwide. Okay? So there was this huge impact, about 120 miles across. It did its thing. You can actually see the immediate impact zone. There was a massive tsunami, things like that. So, extinctions, they clear the decks. You have all these species and a big tidal wave comes across and everything's wiped away, maybe a few things hang on. And then that low biodiversity may persist for a long time, but eventually the remaining taxa, the ones that made it through, are the ones that then start filling up all the niches again. So that's one way you get adaptive radiations. Here's adaptive radiations of mammals. You have Australia. So these are the gray wolf and this Tasmanian wolf. They're not related to each other whatsoever, but they're convergent. They occupy the same niche. They're both big predators. Tree sloth and a koala. Now, all the ones in the Americas are placental mammals. The ones in Australia are marsupial mammals. So you have all these convergent taxa, but in Australia they're all marsupials. And in the Americas, they're all placental mammals. But there's still all these niches that are similar niches, so there's convergent evolution going on there. So here's, again, ecological opportunity. You have dispersal, let's say, to an archipelago, a bunch of islands. There's no competition there. You can have these taxa speciate because there's all these different habitat types. And then finally, okay. And then key innovations, right? Insulation, display, an early kind of dinosaur-like bird, then for flight. Okay. That later. Oh, okay. Real quickly, human evolution. Initially, it was thought that modern humans arose in Europe because, Europeans thought they were the best. Actually, modern humans split up from chimpanzee, an ancestor with chimpanzees 7 million years ago in Africa. They then had separate evolutionary lineages. So, one question was brain size. Which came first, bipedalism or brain size? Well, you can look at the fossil record. They started standing up way before they increased brain. You think intelligence is a great thing. It's really, really, really costly. This little organ up in my head here takes 20% of all the energy that I have. So it's expensive. In other words, intelligence better make that expense worth it. Don't worry. Okay. Neanderthals and modern humans look different, but they actually interbred. They had babies, not very many of them. In fact, the males might have been, the male hybrids were probably not very fit. Okay. Then there was another group that shared a most recent common ancestor called the Denisovans, and they split off about 400 million years ago with these two other lineages. Okay, so this is archaic Homo, and there's these different places, seen realms. Neanderthals were kind of in the west. Denisovans, we don't know really where they all were. And then initially there was modern humans arose in Ethiopia and then spread out through the Middle East. So here's 3 different hypotheses about how modern humans, anatomically modern humans, originated. There's this multi-regional hypothesis where different similar species spread out to different realms, different places, Asia, Europe, Australia, all that. They evolved in place into what we consider modern humans. It's not a very parsimonious explanation. Then there's the Africa hypothesis, where they originated about 200,000 years ago, and then in successive dispersal waves went out of Africa through the Middle East, then into Asia and Europe, and also along the Indian Ocean. And then this one, there's actually no, we can start actually, yeah. There's no gene flow, okay? It was, these guys replaced Neanderthals already there. In this other one, there was some limited gene flow. In other words, Neanderthals mated with humans, okay? And in fact, a lot of us in here have Neanderthal in us. So if you want to kind of, channel your real Neanderthal, you can do that. Okay, I think we should probably call that a day, okay? And here's the test. Thanks, guys. Yeah. Yeah. Enjoy. Yeah. Yeah, she's down. Might make it all the way to the end. You can doodle on these. Okay? Make sure you put your name on it. So that if we, let's say something happens, we have to reconstitute things, I'll at least have 2 different records. But make sure on your Scantron you put which test you're actually doing. There's midterm 1A, 1B, and 1C. They're different colors. 1A is white, 1B is blue, and 1C is green.